Let's take a quick overview of Chapter 13, which covers microbes, fungi, and protists. How do we split up these major groups? One of the things we do when we're splitting up these major sections of the tree of life is we ask whether or not it's a prokaryote or a eukaryote. What that means is, do they have a nucleus? Eukaryote means true nucleus. Prokaryote is before the nucleus. Prokaryotic cells do not have a nucleus. Everything in the blue and red is prokaryote. Another way that we can look at it is whether or not they're multi versus single-celled. I have a question mark here because this one is a little bit tricky, but for the most part, protists are more or less single-celled organisms. Whereas if we look at plants, fungi, and animals, those typically are multicellular. The other thing we look at is a cell wall present? And if so, what is it made of? Everybody in these two groups will have cell walls. The difference is, what is it made of? This area over here in blue will have a different type of cell wall than this whole area here in red. We also see cell walls in fungi, composed of something different. We also see cell walls in plants. Once again, different composition. Next, we can look at the mode of nutrition. Are you an autotroph, meaning you are able to create your own energy using inorganic compounds? You will find autotrophs, of course, in the plants. You'll find some autotrophs here in our protists. If we look at animals and fungi, those are heterotrophs. Animals ingest their foods. Fungi are heterotrophs, but they absorb their food. Rather than bringing the food in and then breaking it down, they secrete their enzymes on the outside and then they absorb that across their body. These are the important things you really have to know where to place animals along this tree. Let's take a look at the tree of life here and look at the groups that we're going to talk about in this video. So these are our prokaryotes. Within the prokaryotes, we have the red group there are the archaea, or sometimes called archaebacteria. Where you see them placed, they actually are a little bit closer to the animals, plants, and fungi because of some of their other similarities, which we won't go into. Then we have the bacteria, sometimes referred to as the eubacteria. We have two different groups here of prokaryotes, archaea and bacteria. In this video, we'll also talk about the protist, so we can see this protist grouping over here. And then we'll also look at the fungi. Early life on Earth was hard. The atmosphere did not have any free oxygen in it. It was anoxic. It means that early life forms had to be able to do anaerobic respiration. Anaerobic means in the absence of oxygen. They had to be able to perform metabolism without using oxygen. It wasn't until later that photosynthesis and aerobic metabolism, which means in the presence of oxygen, evolved. Another thing about early atmosphere was there was a lot of strong radiation. Early life forms were what we call extremophiles. They lived in these really extreme environments, and they still do in many of those groups. Deep ocean floor where there's hydrothermal vents below the Earth's surface, because they were essentially avoiding that strong radiation. Part of what occurs when you have this challenging life is you see the formation of biofilms. And essentially it's like a little community that forms of these microbes, and they interact with one another and they essentially help one another in a lot of ways. So there's a lot of mutualism going on there. Biofilms have become very important because in, let's say, hospital equipment, biofilms will form on the surface of some of this equipment, and it could be very difficult to clean equipment that has biofilm on it. So it's something that is actually very relevant in today's world, not just in early Earth. Bacteria come in three basic shapes. We have the round shape here is called coccus, rod shape is called bacillus, and the spiral shape is called spirillus. And then you also can get them either in individual units or they can form chains. So if you see something called streptococcus, it means it's a chain of these spherical cells. Now, a prokaryotic cell will all have certain features. They will all have a plasma membrane, which is the same as the cell membrane right here. On the inside of that, you'll have sort of this semi-solid, semi-fluid type of a consistency called the cytoplasm. Inside, you'll have a genetic material. Remember, this genetic material doesn't have a nucleus around it. And then you also will have these ribosomes. Ribosomes are for protein synthesis. And ribosomes are a little different than some of our other cellular organelles because they aren't membrane bound. So those are the general characteristics that prokaryotic cells will have. And then they'll typically also have a cell wall. Depending on the type, it'll be a slightly different cell wall. Remember we said cell walls are important. We break the tree of life down into do they or do they not have a cell wall, and then if they do, what's it made of? For the bacteria, there's two different types of makeup of the cell wall. They both have peptidoglycan. So one of the key features is they have a cell wall made of peptidoglycan, whereas the archaea have cell walls that are made of a variety of different compounds, but not made of specifically this peptidoglycan. And then we can separate the types of bacteria into the structure of their cell wall. And based on this structure, they will stain differently when you do a gram stain. So if you have gram-positive bacteria, the peptidoglycan is all here on the outside, whereas over here in gram-negative, this peptidoglycan is sandwiched in here between an inner and an outer membrane. This one stains negative, it's a gram-negative, and then this one stains positive, gram-positive. So it's where that peptidoglycan is sitting that makes the difference.